Hi, now let's continue to study central dogma of molecular biology and the genetic code. So the question for us is how to synthesize a protein from the sequence of nucleotide in a given gene. So there comes a central dogma of molecular biology. So from what we know is from DNA as a template, we can transcribe that information into RNA and translate that information into a protein. So in general, we talk about gene expression. And what is gene expression, by the way? It is the entire sequence of an event that occurs in converting the DNA sequence, which resides somewhere on one of the chromosomes inside the cell into a functional protein. So that's gene expression when we call. So let's see what happens. So this is chromosomal DNA, which is double-stranded helical structure. And if we unzip it, you can see this complementary information of nucleotides. And transcription means based on this DNA as a template, and we can have a complementary RNA sequence here, GC, CG, CG, and AU, like this way. And this will generate an RNA transcript. And we process it into a messenger RNA called mRNA transcript. And this is composed of these also nucleotide sequence. And you can see we lump some as a 3 of these nucleotide. Each will produce a single amino acid. And this we translate. So we call it translation. And this will form a growing polypeptide chain, such as methionine, glycine, and blah, 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 this. So this simple form of central dogma of molecular biology is a little more detailed. It's a DNA transcribed into an RNA. And that original RNA, we actually do some processing of this, especially taking out introns inside and rejoin the exons together. And that process we call splice, meaning like join together. And those processed RNA we call mRNA. And this mRNA is used as for producing protein by translation. So this gene transcription is an RNA synthesis by making RNA. So they're going to be polymerase, which is RNA polymerase. And mRNA, we call it and mRNA is after processing of the RNA, so it contains a message. So we call it as a messenger RNA. That is the result of transcription after rejoining together those exon regions. So we call it after splicing. And there are other kinds of RNA molecules, such as ribosomal RNA, where translation occurs. And there is also transfer RNA. So this is an adapter molecule. So we heard that DNA polymerase for DNA replication, RNA polymerase for gene transcription. So let's compare RNA pol versus DNA polymerase. So DNA polymerase requires a primary regions of DNA to initiate DNA synthesis, while RNA polymerase binds to a region of DNA template called the promoter region. So now, in a nutshell, the central dogma is a segment of DNA which encodes for a gene is transcribed into RNA by RNA polymerase. The resulting RNA transcript is translated into a polypeptide chain based on the genetic code. And that genetic code is related 3-letter. The name is based on this code. So the name is called codons to amino acids. And this leads to the generation of functional proteins. So let's get into central dogma of molecular biology a little further. So the model of information flow within the biological system was proposed by Francis Crick in 1958 in a conference in 1970 in a Nature. So that reference, Crick draw this figure of DNA, RNA, and protein. What is the information flow? So for example, DNA can replicate itself, and DNA we transcribe into the RNA. And RNA can also replicate itself, such as in the RNA virus. And however, RNA can be translated into functional protein. So that's a typical general information transfer. So if I draw it again, redraw it this way as a general, but there are special cases. So we can ask question, hey, if DNA and RNA has a complementary information, why not RNA to DNA? What's wrong with that? So that question is a million-dollar question. In fact, in the nature that exists. So that is called the RNA to DNA, which is the reverse of transcription. So we call it as reverse transcription. And in nature, it actually happens. So let me give you an example of such a devastating immunodeficient disease called HIV, human immunodeficiency virus. This is very hard to treat because this virus is an RNA virus. RNA by nature is very unstable, which means there can be more chance for mutation, which makes it harder to target. However, this HIV virus will infect into our cells, especially immune cells, and they contain genetic material as a RNA. So when they get into the cell, they want to put their genetic information into our genome in DNA. So they actually carry, this virus carry a special enzyme called reverse transcriptase, which makes actually RNA into the DNA so that they put this DNA into our genome, which can be expressed into our cells to produce multiple HIV virus particles. So by this discovery, Howard Kamin and David Baltimore, who got Nobel Prize. So you always ask questions about this, generally accepted phenomena, which is a good attitude. And another RNA to RNA is a RNA replication, which is done by many RNA-based virus. And also, how about, hey, DNA into directly protein? We haven't found it in nature, but direct translation has been happened only in a test tube. While the opposite way, hey, whether protein to DNA, protein to RNA, or protein can create another protein, which is unknown. So let's review the process again. So there is a double-strand DNA, and to replicate, you need a DNA polymerase. While based on this DNA as a template, we create an RNA that requires RNA polymerase, and we call it transcription. And based on this messenger RNA transcript, we actually translate into a protein that's done in ribosome. So let's see inside the cell. Where does this happen? Where does this RNA processing and mRNA translation is happening? First, we have to look at the structure of RNA first. So from DNA, there are promoter regions for transcription. And in fact, not all segment of DNA is actually a coding region. So there are regions which is not involved in direct coding that we call as like intron, because it's inserted probably. And the rest part is called exon, which is a coding region. So once we transcribe, then what we get the original RNA looks like this: exon number 1, intron, exon number 2, intron, like this. And so intron is in fact not necessary for translation. So we actually take this on apart and then join these exons together. So joining is called splicing. So we call it as a splicing. And this part is part of RNA processing. So this contains the coding information. So we call it a messenger RNA transcript. And after translation, we can create a polypeptide chain or protein. So these coding regions of DNA we call exons. And while non-coding regions, we call it introns. So primary RNA transcripts undergoes splicing to remove unnecessary introns. And let's see how this happens inside a cell. So this is cell double membrane, and this is a nucleus, and inside there's a chromosome, and inside DNA. And DNA first make RNA transcript. Transcription makes an mRNA, and that mature mRNA will come out of this nucleus and transport it to cytoplasm. And inside the cytoplasm, there exists a ribosome. And this single strand messenger RNA transcript is now ready for producing a polypeptide chain by translation. And that scientists consider, how does this happen? We do need some adapter molecule which corresponds these 3-letter information into one single amino acid. So there comes an idea of a transfer RNA. And that is happening at this complex machinery of ribosome. So this nucleus containing eukaryotic gene, there is a promoter region containing binding site for general transcription enzymes and other transcription factors. And the introns are spliced and removed to yield an mRNA transcript, which consists only exons. And the resultant mRNA is then translated into a protein. Oh yeah, so this translation occurs at the ribosome. So now thinking about the genetic code, how to convert a linear sequence of nucleotide in mRNA, which is composed of A, U, C, G of 4 types, into a linear sequence of amino acid, which is building blocks of a protein. And we know there's about 20 amino acids. So that is having this mRNA transcript, which also has a 5' end to the 3' end. And interestingly, now let's take it because we will discuss more later. So this 3-letter of nucleotide corresponds to 1 amino acid. Then this transfer RNA should be adapting this. So one side, such as, let's say, there's a GUA, is supposed to produce a valine. Then there's a complementary sequence C, A, U. And one side is, and the other side must have this valine amino acid, and they will attach to this as a growing polypeptide chain. So this polypeptide synthesis is that amino acids are added to a growing polypeptide chain by tRNA. This tRNA serves as an adapter molecule, and each tRNA should contain the opposite of this codon, so we call it as a anticodon, so which is complementary 3 letters on one end, and the other side, the corresponding amino acids should be attached. So 3 mRNA bases we call triplet or codon. This is read at one time by an incoming tRNA. So if the anticodon of tRNA is complementary to the codon region in the mRNA, then amino acid from that tRNA is added to this polypeptide chain. So now you will have a question like, hey, why is the codon made up of 3 amino acids? In fact, in history, figuring out this actually led to Nobel Prize again. So this is called finding a genetic code. And you see from this, based on the mRNA, so there's 4 types of nucleotide: uracil, cytosine, adenine, and guanine. And so the first part is this, and the second letter can be also UCAG, and the third letter is UCAG. Then how many of these 3-letter sequence can create, which is 4 times 4 times 4, which is 4 to the 3 or 64. And what is the final number of amino acid, which is 20. So, you know, the genetic code is actually not like exact one-to-one, but any 20 amino acid can come from maybe several different mRNA sequences. And this finding and this genetic code, we call it that nature as a degenerate, only used in biology as the term, the ability of elements that are structurally different to perform the same function or yielding the same output, called degenerate. So let's think a little further about the genetic code. How many nucleotides of mRNA are required to encode the 20 amino acids? And we said 3 bases along the mRNA chain for 1 amino acid is called codon. That's a minimum unit of genetic code. And interestingly, there are codons which is leading as a start codon and stop codon. So you can see here, like these 2 UUC will code for phenylalanine, and these will code leucine, and AUG will encode methionine. And you can see there's like stop codon, stop codon, stop codon. And this methionine AUG is in fact start codon. So what it means is the translation event starts at this start codon. So it always starts from AUG on mRNA, and it terminates when it sees the stop codon. And interestingly, there's a 1 to 3 stop codon. First is UAG and UGA, UAA. And so if I want to summarize the transcription and translation by comparison, so transcription starts on DNA segment, which is called promoter region, and then it stops at the terminator region. While translation is based on mRNA, and where it's AUG or called start codon, and the other UAA, UAG, UGA as a stop codon, which means translation stops there. And the degeneracy, as I alluded, any 20 amino acid can come from several different mRNA sequences. I'm going to discuss this one further.